In this activity, you're going to explore how plant roots form symbiotic relationships with other organisms, specifically bacteria and fungi. These partnerships are incredibly important in natural and agricultural systems because they help plants obtain nutrients more efficiently than they could on their own. You'll look at two major examples: root nodules, formed with nitrogen-fixing bacteria, and mycorrhizae, which are associations between fungi and roots. A symbiosis is a close association between two different species. These relationships can take several forms. In mutualism, both partners benefit. In parasitism, one benefits while harming the other. In commensalism, one benefits and the other is essentially unaffected. In plant roots, we're focusing on mutualistic relationships, partnerships where both the plant and the microbe gain something valuable. One important root symbiosis involves root nodules and nitrogen-fixing bacteria, usually species of rhizobium. These bacteria colonize the roots of certain plants, especially legumes like alfalfa. Inside the nodules, the bacteria convert atmospheric nitrogen, or N2, into ammonia, NH3, which the plant can use to make proteins and other important molecules. In return, the plant supplies the bacteria with sugars and a protected place to live. From the outside, these nodules appear as swellings on the root surface, but inside they're packed with bacterial cells. Another extremely common root symbiosis is mycorrhizae, which are associations between fungi and plant roots. The plant benefits because the fungal hyphae greatly increase the effective surface area of the root system, improving water and phosphorus absorption. The fungus benefits by receiving sugars and other nutrients from the plant. In most ecosystems, the majority of plants form some type of mycorrhizal association. There are two types of mycorrhizae you'll be looking at: ectomycorrhizae and endomycorrhizae. In ectomycorrhizae, the fungal hyphae form a sheath around the root and extend into the outer cortex, but they do not enter the cortex cells themselves. In endomycorrhizae, the fungal hyphae actually penetrate into the cortex cells. They grow inside the cells without breaking the cell membrane and can fill much of the cell's interior. Before you begin your observations, think about this question: why might plants form partnerships with bacteria or fungi instead of relying only on their own roots for nutrient uptake? Consider things like efficiency, surface area, and access to nutrients in the soil. Write your prediction in your lab notebook before continuing. First, observe the plant with exposed root nodules. Identify the nodules as the swollen structures on the root surface and think about where your bacteria are living inside the root. In your lab notebook, sketch a root and label one nodule. This will help you connect what you see on the outside to what's happening inside the root tissue. Next, look at the prepared slide of ectomycorrhizae. Under low power, focus on where most of the fungal tissue is located relative to the root. Then look at the higher power and observe how far into the cortex the fungal hyphae extend. Remember, in ectomycorrhizae, the hyphae do not enter the cortex cells themselves. Sketch what you see in your notebook and label the hyphae and the cortex. Now examine the prepared slide of endomycorrhizae. Under low power, check whether you see fungal structures on the root's outer surface. Under high power, look carefully to see whether hyphae are present between cortex cells and inside the cells. Estimate how much of the cell's cytoplasm is filled with fungal hyphae. Sketch this association in your notebook and label the hyphae and the cortex cells. Now compare what you've seen. First, think about how the plant benefits from both root nodules and mycorrhizae. Even though one involves bacteria and the other involves fungi, what do these partnerships have in common from the plant's point of view? Second, compare the location and appearance of endomycorrhizae and ectomycorrhizae. Focus on where the fungal hyphae are located relative to the cortex and whether they enter the cells or stay outside. To wrap up, root symbioses are powerful examples of mutualism. In root nodules, bacteria provide usable nitrogen to the plant. In mycorrhizae, fungi improve the plant's ability to absorb water and phosphorus. Ectomycorrhizae stay outside the cortex cells while endomycorrhizae grow inside them. In both cases, these partnerships make plants more successful than they would be on their own. Once again, this shows how structure, function, and interaction with other organisms all come together in plant biology.